All right. Good morning, good afternoon, good evening, wherever you're joining us from today. Thank you for joining us for this webinar. I think we'll start. Other people can join us as we go. So today we're going to be talking about how to make the right decisions for your library preparation, because successful sequencing begins long before your material reaches the sequencer. So today we want to explore what are those factors that influence your decisions and what are the right choices and the best practices for you to achieve a great library so that you can get your good data for the work that you're doing. So joining us today for this session, we're joined by Charles Wairure, who's a senior FAS at Illumina, and let him introduce himself after I introduce myself. So I'm Cecilia Rumberia. I'm an application specialist at F&S Scientific, and we work with many of you who are joining us today, and some of you might be meeting us for the first time. I basically am involved in supporting the NGS workflows end-to-end with individuals who are practicing on the bench, basically doing genomics work. So all the way from sample preparation to library preparation, sequencing, as well as data analysis. And I also work with Dr. Peter Npiga, who also supports the NGS workflow. So I'll allow Charles to say hello and maybe introduce himself, then we can proceed. So Charles, over to you.
Thank you. Good morning, everyone. As introduced, I'm Charles Kamau, a Kenyan based in South Africa. I'm a senior field application scientist for Illumina. It's actually just 2 years since I joined Illumina, but I'm not that new in the industry because prior to joining Illumina, I have represented Illumina through a channel partner like FNS, but based in South Africa. So similar to what Cecilia is doing in Kenya and my other colleagues, we're involved in making sure that once you make the right decision in purchasing the platform or the library prep kit or the array platform, and then once you get that, with the one who are involved in making sure that you use it the best way possible through the trainings we receive. And you'll get to know me more as we carry on because I have more to share with you and I hope you'll find the session insightful. Over to you, Cecilia.
All right, thank you. So today, Charles and I will drive the conversation. And on the sidelines, we'll be supported by both Dr. Nancy, who's also a FAS at Illumina, and Dr. Peter Npiga. And with me also, I don't work alone. I have a team that might be able to answer some of the questions that you have around sample preparation, quality analysis, and such. So we like to make this interactive. So do feel free to put questions in the chat, to share what you might be working on in case you'd like us to maybe discuss it and offer inputs for something that might need some questions answered. Yeah. So I'll begin with introducing Illumina for the sake of those who, just briefly, those who might not know who Illumina is. Illumina is a company that has been around for many decades, and I think Charles will do a better job at introducing Illumina. But the mission of Illumina is to improve human health by unlocking the power of the genome. And we all hopefully know what the genome is. Essentially, it's the genetic code of an organism. And in this case, for Illumina, it's about the human health. So our conversation today is around making the right library preparation decisions. And we're gonna go over where does library prep fit in? How do we make the right decisions for our workflows? What are the best practices? What are the pitfalls that lead us to having a library that doesn't give us the quality of data that we would like? And then we look at what questions you might have, either based on the presentation or based on what you're already experiencing right now. And then community perspectives, maybe what users have experienced and what advice they could give to us all here today, because we all experience different things when we get on the bench. And being the scientists that we are, we kind of know how to troubleshoot and finally get there. So hopefully we'll also get to learn a few of the troubleshooting and adaptations that you've done to workflows. So where does library preparation fit in? First of all, maybe I'd like for us to go over, when we talk about unlocking the power of the genome, essentially we mean unlocking the genetic code. It could be of the human or of another organism that is of interest to you, whatever you're studying. And the reasons for this could be varied. So someone could be studying to understand an unknown species, maybe for discovery or for characterization. So in this case, you might be looking at sequencing like the whole genome of an organism, or understanding how the organism functions. So maybe in terms of genomics, the transcriptomics. Your mission could be biosurveillance, which is maybe identifying pathogens within an ecosystem or within a system or a patient. Another reason would be mapping genomic structures. How do different genes interact? Studying variations and evolution, looking at how genes influence phenotypes. And today we even have genomics for targeted therapies. So a lot of the questions around which library you're doing will be towards what is your objective. The reason for NGS essentially is why we have the research questions or the clinical questions. And part of this workflow includes the library preparation aspect. That is why we want to discuss it in detail. And there are various approaches to answering your research questions. As I've mentioned some of them, it could be that you're looking to do a whole genome sequence of a pathogen, of an organism, a bacteria, virus, or even of human. You could be looking at understanding the expression pathways in maybe a disease status. So RNA sequencing, which could be whole exome, or even total RNA sequencing. We also have now a lot of work around single cell sequencing. This is looking at the immunology status, like in disease responses, or how tumors are functioning within that the space of the tumor, for example. There are also workflows around DNA methylation. DNA methylation occurs and it impacts how the genes are expressed. So silencing of genes occurs. So understanding which parts of the genome are methylated could give us knowledge around how is the gene being expressed, or how is the gene actually functioning. Then we have metagenomics approaches, either targeted or shotgun. Shotgun is where you don't quite know the organism or the DNA that you're looking for. So you want to sequence everything within your sample. So this allows you to tell you which organisms exist within that sample, be it bacteria, parasites, the host material. The targeting where you already know that maybe you're studying like bacteria. So here you'd go for an approach like 16S sequencing, where you first amplify a specific region and then you sequence it. And that would also fall within the targeted amplification sequencing. And let me just grab a laser here. So the targeted gene amplification, where you could amplify a specific segment, like assuming you're studying maybe oncology, you could be looking at changes in a specific gene, like the BRCA gene, the T53 gene, to understand a research question that you're studying. Now, library prep is only part of the workflow. A lot happens before library preparation, and a bit happens after library preparation before you get into the sequencer. So one, of course, you start with obtaining your sample. These samples are a variety. There are a variety of samples. It could be blood, swabs, tissue, insects, depending on what you actually focus on. So extraction is the first part of obtaining either your DNA or your RNA. And this can be either you're doing manual extraction or you're doing automated extraction. And the idea is to obtain a good quality DNA and RNA. Now, because sample sources are really varied, you could be working with a sample that is really difficult to work with. It could be maybe a leaf sample that ends up giving you a lot of phenolic compounds, or you could have a small sample. So maybe your DNA or RNA concentration might end up being little. So all these factors downstream are part of the workflow and affect eventually what kind of data you get. But Charles will go more into details around the impacts of the different qualities. So library preparation is where now you prepare your sample to be ready for the platform that you're going to be using, for it to be compatible to the sequencer so that we can be able to now read the genetic material for downstream interpretation in analysis. The sequencing part also varies quite a bit because there are a number of sequencing platforms in terms of scale. So if you have a small project, you would optimally choose a sequencer that allows you to optimize the cost as well as the data output that you get. Now, all these are interlinked, and that's why today we really want to focus on the library prep after aspect. As I've mentioned, one, sequencing data quality is directly related to your library quality. Whatever you load on your sequencer will be reflected by the data that you get. If your library quality is not great, then you will not get great data. If your library quality is good, then obviously you will get good data. And library quality is determined by everything that happens before, during, and after the preparation process. So when I say before, I mean the starting sample quality. Sometimes we don't have the direct influence on the quality that we get. I'll give an example of maybe FFPE samples that have been fixed for histology. You end up having maybe a very small amount of sample to work with, and maybe a sample that will result in highly fragmented DNA or RNA. But there are ways to select a library workflow that will allow you to work with this quality of sample. So the question when you're beginning is, what kind of sample am I working with? Two, which method of extraction are you working with? Is it going to allow you to get a good quality DNA or RNA that you can work with, and a good quantity of DNA or RNA based on the library prep workflow that you will be working with? Then during, now, depending on the library selection that you go for, you might need to start with fragmentation. So that, of course, impacts the quality of library that you'll end up with. Do you fragment, under fragment, over fragment, or optimally fragment your library? Then some of the workflows include a PCR step, which increases the concentration of your library. Some do not include a PCR step. It really depends on the objective and the workflow that you select. Then the steps for indexing and adapter ligation, you need to be able to do a good job, either ligating or attaching your adapter, whether it's a PCR-based or a ligation-based workflow. If this is not done correctly, again, it will impact on the quality of your library and then impact on the data quality of your sequencing. Size selection in terms of the library size is also a very critical step in the cleanup stages, because you need to get rid of primer dimers and adapter dimers before you load your library into the system. Operator technique, of course, is important. You need to do your workflow very well and organize your work in such a way that you're not skipping steps, you're adhering to the protocol or the guide as it is. And if you make any changes, they better be optimized to work, because I know there are obviously times where we adjust protocols, maybe to have a customized workflow based on the research questions, but those impact on the library. They can improve the quality or reduce the quality that you get. After your library is ready, you have gone through the workflow, you have a ready library, and you have done your QC. Okay, no, the next step is you do your QC. That is a quality checks. One is quantification. You need to do this correctly, either using fluorometric methods. Those are the recommended methods to be able to know how much concentration do I have. Because downstream, we will be loading the sample into the sequencer, and there are very defined metrics around what concentration you load into your sequencing reagents and workflow. Two, there's fragment analysis. What is the size of your library in terms of the average distribution? When you're selecting your sequencing cartridge, for example, if you have a very long library, okay, yes, long, and then you select a workflow that does maybe short read, 300 cycles, 100 cycles, and you have a large library, that will impact on the quality of your data. Indexing, I bring it up here again because we end up creating a sample sheet for the instrument to be able to demultiplex your samples. So you need to have a log of how did you index your samples if you're multiplexing a large number of, or even a small number of samples. As long as multiplexing is happening, the indexing has to be done in such a way that you've selected diverse indexes, not too closely related, because there'll be conflicts when the software is trying to call the different indexes for demultiplexing. So this is also very key in logging the correct indexes alongside the right samples. Size selection is part of library prep, really, and storage and handling of the library itself. Assuming you've created your library, it is ready, but you're not yet ready to sequence today, how do you store that library? How do you store it after? And then how do you normalize, dilute, and pull to be able to load it onto the sequencer? All these are aspects that will influence the outcome downstream. Now, for nucleic acid extraction, one of the things that is very important is ensuring that you do not have contaminants in your sample and you have the right starting concentration based on the workflow that you're going to be utilizing. Many of us, I think, in our labs already have a nanodrop, which is a very good QC for contamination. But again, I've mentioned the best workflow for doing the quantification to get exact concentration would be from fluorometric assays. But nanodrop always does help to check that contaminant profile so that you know if you need to run a purification step to improve the quality, to reduce the contaminants in your sample before you get into library preparation. There are very many options of library preparation. After you've determined that your DNA or your RNA is good concentration, it's good quality, it's good enough to go down into library prep, you could select workflows that are PCR free. The main reason for that is that there's always a bias with PCR. So if you do not want to introduce any bias in terms of what you're sequencing, then you essentially would pick a PCR-free workflow, which essentially means that you fragment your DNA. Illumina now has solutions that allow you to fragment your DNA enzymatically. I think when NGS started, I think with the earlier systems, we'd need to fragment our DNAs manually. So with instruments, sonication or hydroshare instruments, but now we have systems that allow us to fragment with an enzyme, which creates that uniformity in the sizes that you end up getting. So once you fragment and you're choosing a PCR-free workflow, you ligate your adapters. So ligating adapters will allow you to be able to utilize the flow cell, and also indexes will allow you to be able to multiplex your samples. And you can also select a workflow that is PCR, that has a PCR amplification step, for example, Illumina preparation, Illumina DNA prep. So this one still involves the fragmentation enzymatically, but then now how you add your adapters includes a PCR step, which increases the concentration of your library and improves the amount of library that you have. Another workflow structure is where you do the Illumina DNA prep, as I've described here, where you fragment your DNA and segment your DNA, add adapters and indexes using a PCR stage. And then after your library is ready, assuming you have maybe a patient clinical sample and you only want to enrich for a specific pathogen, maybe a virus or a bacteria, then you use hybridization probes to fish out those specific library molecules that you want to sequence. So you leave out everything else, like the host library components, other pathogen components, you only sequence what you're interested in. So those are three options that I'm highlighting here today. And as I have mentioned, a good library essentially will give you a good data outcome. You can do your library all the way from beginning to end and do your QC, and you see I have a great library. It is good concentration. It has the right fragment distribution that I want. But you also have to be keen on how you handle your post-library preparation, how you normalize, dilute, pool, and load into the sequencer, because that can also affect the outcomes that you get. So what we are hoping to achieve is to at least you understand, one, your sample source influences the decisions that you make around how will I extract my sample, in terms of maybe the size of sample that you'll extract, how much will I elute in, how do I quantify and check the quality, and then how do I select the library prep workflow that allows me to answer my research questions, how do I QC to make sure that I'm getting it right at the different stages, and then how do I prepare my library for the sequencer. So a sequencing run, as long as you load a poor quality library, you will get poor quality data set. So it is very important to do the QC stages and make sure you're running the right library. So sampling, of course, you collect the right sample in the right quantities, or you investigate, what am I working with? What do I have in my hand? What am I starting with in terms of sample? Sample storage, of course, is very critical. How you store your sample, how you ship your sample will affect the quality of your sample. Extraction, there are various options, and you're not limited to manual workflows. You can also run automated workflows. This is usually influenced by the number of samples that you're handling. If you're handling too many samples, like your lab routinely does a lot of samples, there are automation solutions that allow you to standardize the workflows so that you get consistently standardized DNA or RNA concentrations or outcomes. Library prep, I've mentioned, there are various options, again, depending on your research question. There's also manual workflows for your library, and there are automation solutions. If you consistently, again, do a lot of libraries, might be worthwhile to think about an automated system that allows you to also reduce the operator subjectiveness or variation when it comes to the quality of libraries that you get. Library QC is a must. If you load your library into the sequencer without checking what quality you get, it will be reflected in the data that you get. There are a number of sequencing platforms that exist within the Illumina portfolio, and the one that you use depends on how much data do you really want and how many samples are you working with. If you're just you could be working with 2 samples that have a small genome and you're doing whole genome sequencing, then the low throughput systems would be sufficient. In the same breath, you could be doing whole genome sequencing of 2 samples, but they are human samples or they are huge genomes. Then you will need a larger throughput instrument. Beyond the sequencing, now you have your great data. You need to do the analysis. There are solutions that exist that are automated, or you could also work with custom pipelines if you know how to code and build scripts and build bioinformatics pipelines. So the entire chain from sampling to the kind of data that you get and the kind of insights you report or you utilize for your work are determined. You have to make decisions around each stage, because at the end of the day, what you want is great data and insights that are usable. And I'll stop there and hand it over to Charles. Maybe Charles, you can share your screen and take us to the next stage. Thank you.
Okay, thank you. Let me share my screen. Let me know if you can see my screen or not yet. Okay, can you see my screen now?
Yes, we can see your screen, Charles. Thank you.
Yeah, thank you. So for the sake of those who have joined late, let me start with an introduction. So I'm Charles Kamau Wairuri from Kenya, and I'm a senior field application scientist for Illumina based in Johannesburg. And today, following up the introduction from Cecilia, I'm going to be giving you a practical framework to ensure that you have successful NGS projects, mainly around selecting the right library kit. As you know, there's so many options out there. Some are Illumina, others are from other market players because our platforms are open. But I want to give you a foundation on the right way to choose the right kit for your application. But before I get there, I would like to introduce you to Illumina, who we are. We are known as a global genomic and human health leader in driving precision health. We develop, obviously, sequencing platform. That's what we are well known for. But we also do microarray, bead-based array. And we have this being applied in several industries, be it in oncology, genetic research, agriculture, infectious disease, and I'm sure some of you represent those industries as well, including the reproductive health as well. Okay, so Illumina is not a small company. As of 2024, we were valued at about 4.3 billion US dollar. That's a lot of money. Please don't try to convert it to our local currencies. Yeah, we have a big number of employees, now more than 9,000. And we are now with our third CEO, Jacob Tyson, and our headquarters is based in San Diego, USA. So we have been around for a very long time. We started way back in 1998. So we have been around for just over a quarter of a century, so more than 25 years. And as you know, out there, we ship more than 20,000 sequencers globally. That's a big number. And the publication from way back when we started the HiSeq and obviously the MySeq, which is coming to end of life, we have more than 300,000 peer-reviewed publications. So we're doing some good work, and I'm happy and honored to represent Illumina and seeing how we can indeed help improve human health by unlocking the power of the genome. Okay, so where are we? We are everywhere, but physically, I'm happy to report that, now we even have a commercial office in Johannesburg, and that's where I'm based. But we have other representation across the globe as well, as you can see from the map we have. As I mentioned, we have now Illumina Africa, where we have been involved in several projects. Like since 2003, we were among the leaders in fighting Ebola outbreaks, obviously COVID-19, Mpox, and recently we had the hantavirus. And we operate in 52 out of the 54 African countries. But obviously, you all fully understand, because I know we have representation across the, Africa. We cannot do this alone. So we do work with a model we call hybrid model, where we have currently have 11 channel partners. And as I mentioned earlier, although I'm only 2 years with Illumina, I've been representing Illumina for more than 10 years now, because prior to this, I worked for other channel partners like Whitehead Scientific, who are distributing for Illumina product, and more recently, Separation South Africa, before I moved on now to work for Illumina directly. And in Africa, we are about 14 employees. And I think also in the audience here, some of my colleagues are there. We have Nancy representing East Africa. We have Uzoma representing West Africa. And those are just application specialists, but we do have commercial and the engineers as well. And we recently just hired a field application specialist, a French-speaking one, because we know certain countries are not, French-speaking, and we struggle. I have had challenges trying to present a scientific call like this with an interpreter. It's not that easy. So I'm very happy that Sarah has joined us actually last week, and you'll be seeing her activities as well. Okay. So moving forward, you're gonna have some of repetition, and I always say if it's that good, it can be said more than once, And I want to have a little bit more interactive session, and my colleagues will be monitoring on the chat as well. So we just covered few aspects, Cecilia went through few aspects of Illumina sequencing workflow, and I'm curious to see how many people can remember at least maybe all the 4 steps that we do on the platform. If you don't mind, you can put your answer on the chat, and before I proceed, so I'll give you a few seconds and see if you're able to at least remember 1 or 2. Because I'm not able to see the chart, my colleagues, if you see any answers, you can mention to me. If I only get 2 or 3, I'm happy to proceed. Which are the 4 Illumina sequencing steps that we follow? We did cover those this morning, so you should remember that quite quickly.
So we have an answer from Lydia: DNA extraction, library prep, sequencing analysis.
Okay, that's good enough.
Sequencing and analysis, yeah.
Okay.
We have Mboro, extraction, library sequencing analysis.
But you see there is a keyword, Illumina sequencing workflow steps. Anyway, let me stop torturing you. Obviously, as Illumina, we mainly focus on these 4 steps. Obviously, the library preparation, then which is followed by cluster generation, and then sequencing, and then the data analysis, of which for most of the workflow we have, library preparation is the only step that happens outside the instrument, with the exception of the TruePath genome, which we'll talk more about in later session. Okay, and here, obviously, during the library preparation, just basically converting the DNA or the RNA into a molecule that's compatible with our platform. So we add adapters on either side, and those are the steps we follow. And most of the talk now will be revolving around the library preparation. The reason I'm saying TruePath is an exception to this is, as you might just let you know, this is the only workflow we have whereby we actually introduce the DNA, isolated DNA, directly into the flow cell. It's the most comprehensive genome you'll ever get, and it has the easiest steps. So even if you have like a technician who has rusted over the year because they haven't done any pipetting, it's only 5 steps, and then you have the most comprehensive genome you can get, including areas that maybe our typical SBS technology, short technology, was not able to cover, but we'll talk about that more in detail. Then we have cluster generation. You know, prior to the introduction of the MySeq, about 2011, we used to do this step outside the instrument as well. You know, so the HiSeq used to do this outside the instrument, then introduce the clusters, and you carry on with the sequencing. But as you can see, Illumina is continuously innovating. You know, from now having all the platforms we have, the cluster generation happens on board the instrument. So you don't need to do anything as long as you control every aspect in the library preparation and how much you introduce, you sorted. So the instruments are quite robust for that. And they'll also, after doing the cluster generation, if indeed you have any, then you go to the sequencing. And our technology hasn't changed, but we keep improving on it. We started with the SBS sequencing by synthesis, and now our flagship platforms like the MySeq one, Harden XSeq 2K, and NovaSeq X, we now do XLIP SBS, which is an improvement of the old technology. We don't stop there. We do carry on, and we convert the images we take into usable data. So we would start with the primary analysis on the instrument, which we convert the images, because here we just, it's a fancy camera, you know, we're just converting those images we take on every cycle into base calls. But those BCLs means nothing. So we have there now to assign those onto the different samples. That's why we have the FASTQ files. Once you have the FASTQ files, which is the output of a sequencer, then we do the secondary analysis. This may involve the alignment maybe to a reference genome, a manifest for variant detection. Once again, you may have different variants, whether you're working with infectious disease or oncology or whatever research. But what does that really mean? We now have now the tertiary analysis, which is basically making sense of the data you have, you know. And as we mentioned earlier, in as much Illumina doesn't have solutions for sample collection and purification of DNA, RNA, or protein, you know, those are very crucial steps, which were nicely covered by Cecilia earlier on, but they are very critical because you may have the best library kit, the best sequencing platform, but if you introduce the wrong materials, then don't expect miracles, okay? So it's very important that we take into consideration the input we are having. And that's where we have reference guide for almost all the protocols we have, where we'll give you clear guidelines on how much nucleic acid do we need, what integrity, like how should it be degraded? Can this workflow support degraded DNA like what you get from FFPE or any ancient samples, you know? So we need to be careful and follow that one religiously. And if you do that, I can ensure you're going to have successful runs because the instruments are quite robust. Okay. So we keep talking about library preparation, and I just decided, let me give this animation here that helps you explain what a library is, irrespective of the application, including the true path genome, which I said we do library preparation on the instrument. Yeah, all the libraries end up looking exactly the same. If you start with RNA, we have to convert the RNA to cDNA, you know, and then we proceed with the fragmentation. And as previously mentioned, this can be either enzymatic, or you can use like mechanical fragmentation, which is actually how Illumina started before they acquired a company called Epicenter, also like some maybe 10 years ago. We only used to have the TrueSeq kits, which needed you to have a Covaris for the mechanical fragmentation. Yeah, but that's quite expensive instrument. Yes, it does work quite well, but you also require a bucket full of DNA to get started. So most of the workflows we have, we are continuously improving on them, and the enzymatic one are working equally well. That's why you'll see like a lot of the work that's being done now, we use the enzymatic-based fragmentation, okay? And the reason why we are fragmenting, remember, we are sequencing using short-read sequencing technology, yeah? And what we do is, during the library preparation, or what you're aiming to do is, you have your DNA, you fragment it, and then you add the adapter. And the adapter, we have the P5 and the P7. Those are essential for you to, for your library to be able to attach to the flow cell where we do the cluster generation. So should you be missing either P7 or P5, you know, because remember, there are some workflow that add these adapters through ligation. So it's possible you can add P5 and you miss the P7. That molecule may not be able to cluster on a flow cell, and for that, you're not going to be able to get any data out of that. The clustering is going to fail. And that way we have to do those QCs to confirm, indeed, do I have viable molecules? And I'll talk about that one in a later session, okay? Then the beauty of next generation sequencing is the ability to sequence as many samples together. This is what we were missing before. I remember when I was doing Sanger sequencing, you have to purify even the amplicon to make sure that the forward and the reverse primers are not together. Here, we want to mix as many samples as possible to achieve the desired coverage, and that way we have to add the EDEX1 and EDEX2. So you can have one EDEX for a single EDEX strand, but most of the work we do, especially with enzymatic fragmentation, we do dual EDEXing, okay? And as you'll notice, a lot of the platform you're working with is these patented flow cells, whereby we have nanowells where we have the clustering taking place. So we prefer using the unique dual EDEXs, whereby no any sample shares either EDEX1 or EDEX2. Remember, we still have the combinatorial dual EDEXs, but they share the first names, like having a similar surname, if that makes sense. Then what makes this technology as beautiful as it is because you can sequence anything. As long as you can figure a way to add these adapters, which the sequences are literally available that we even have other third-party chemistry being able to create library that are compatible with the Illumina platform, we can sequence that because you have these universal read 1 and read 2 sequencing primer binding sections. You know, every library you'll ever sequence on any Illumina platform, be it on our old, you know, the smallest to the biggest, they all have exactly the same look. It has read 1 and read 2 sequencing primer. Okay. And without, if you have any of these missing, then you have an incomplete library. So to conclude, you need to have the P5, P7, obviously EDEX1 and EDEX2, and the read 1 and read 2 sequencing primers binding sites. Okay. So just to pause a little bit, and ask a few questions. So what are the key components of a good library? We just covered that one. So I only need two or three answers, then we can proceed. And also have, if you can be able to answer both questions, which Illumina workflow performs both library preparation and sequencing within the sequencing consumable, eliminating the need for the conventional off-instrument library preparation. Those are my two questions when I take a break. Any answers? What are the key components of a good library? In which workflow did I introduce that does the library preparation on the sequencing cartridge? Sometimes I listen to some of these things and I'm like, I'm dreaming, but that's the beauty of Illumina. They keep pushing and, expanding new boundaries. Do you have any response?
Not yet.
Come on, did I put my people to sleep?
Well, while you're at it, we do have a question in the chat around TruePath.
Yeah.
So is it a new kit, and how does it compare to the Illumina DNA prep kit, the TruePath genome that you've mentioned?
Okay, so we will have a later session where we'll go into a little bit more detail. So it's a brand new kit, yeah, that's only compatible with the NovaSeq X. And the main difference is we are able to get a more comprehensive genome, which is phased. Remember, as a human, for example, we are diploid, so you have one portion coming from your mother and the other one coming from your dad. So the traditional way of sequencing, that has been a very difficult thing to distinguish. Now we're able to look at the genome that's phased to see, oh, this is what I got from my mom and this is what I got from my dad. Then there are other difficult to sequence genes that traditionally we were struggling to do that, and only long read technology could be able to do that. And few that are out there, some may be good, but they're too expensive. So this one, we use the same technology and we are able to even cover those genes that were or gene region that were very difficult to sequence prior. Okay, and the main point here is it's the easiest workflow you can have, easiest to automate. You only have 5 steps. But as I said, we'll talk about that in more details later on. Okay. So,
all right. We do have an answer on the question that you asked. So P5 index 1, SP1 DNA template, SP2 index 1, and P7.
Yeah, perfect. Okay, thank you.
Thank you, Sam.
So
Samuel.
going back. Sure. They took too long. If you take too long, then I'll assume you're asleep. I hope the next one I'll be less. I'll not put you to sleep. So remember, as we keep saying here, let your biological question guide you to the kit you're gonna be using. So the first thing, you need to have a clearly defined research questions. For example, I want to characterize a genome. I just want to know the blueprint of this organism I'm working with. So the other question I would ask there is, do you want to characterize the entire genome? Is it part of the genome? And if it's a genome, what kind of mutation are you after? Is it somatic mutation, which are localized to certain areas, which you only acquired later? Or is it germline mutation, being that we say probably you inherited this from, it runs through the family? Are you looking at the genome sequence only, or you want to see also if there is any changes to the genome, not necessarily change in the sequence, but changes in modification, like what you have seen with the methylation? Is it the epigenome, whereby you see the cytosine have been methylated? And we know this has an effect in a lot of things, including controlling gene expression, okay? No, I'm just interested in seeing whatever activity I have in this organism. So I'm looking at gene expression. So there you're looking more at RNA. Are you looking at rare disease? These are diseases that they are very unique, so you don't know exactly what to look into. And some of these rare diseases are really taking full advantage of like the true path genome, because we are uncovering areas that before had not been characterized or associated with any disease, you know. So in infectious diseases, am I just looking for pathogen surveillance? We know like we now need to be more alert because we don't know what's going in which country. So regular surveillance can help us, you know, pick a pandemic before it become a full pandemic. Then we can start controlling how people are going to be operating. Am I looking at just microbiome analysis? Okay, these questions will help you define which platform to use, which kits to use, and other questions you can carry on and say like, okay, what materials do I have? Am I working with DNA? Am I working with RNA? Is it proteins? Okay. And then there we can decide if it's RNA sequencing, we have different solutions. Total RNA, where you're looking for both coding and non-coding, or messenger RNA, just the coding region of our genome, a transcriptome. Or am I looking at the microRNA, which we have seen they do have a major effect in gene expression. You can get, looking at our typical central dogma, you can see the DNA is transcribed to RNA. We can pick the RNA signal, but we don't get the protein because something interfered with the translation. And we have seen microRNA being involved in that. That's why a lot of work is done around that, even as for drugs as well. Okay. And with the whole genome, depending on the platform you have, or with the DNA sequencing, sorry, depending on the platform you have, you can decide, okay, I'm going to go for the whole genome sequencing because I have a NovaSeq X, which can do that comfortably, you know, or I have a limitation because I only want to see the coding region of the genome, and that's why we go for the whole exome, or even lower for the small benchtop platforms like our MySeq 100 can go for the targeted panels. And as Cecilia mentioned earlier, you know, you need to evaluate the sample input requirement. Remember, every protocol we have, every kit you're mentioning, we do have a reference guide, and we'll tell you this kit only works with this amount of maybe this high quality DNA or RNA, you know. So you need to make sure that you're choosing the compatible solution for the kit that you're using. We know low input, like the cell-free DNA, you know, like what we do for the NIPT and all that, the single cell and other nitroaspirates. They are very little amounts, so you need to use the right kit, you know. Okay. So all those questions you ask, and obviously what you have within your facility. A lot of people are very keen on, let's say, for example, the true genome, but do they have the sequencer to do that? Luckily, we're now getting more service providers, and I'm happy to see, like in Kenya, we already now have a NovaSeq X Plus. So that means any of the conversation we are having here, if you're in Kenya, we have a solution, we have a center that already have a NovaSeq platform, next gen, and you can actually ask for those services. Okay. So just to bring that conversation together, because we keep saying different, you know, we need to know the genome size. We are about sequencing, you know, or genotyping. So our communication is, what am I sequencing? Is it a virus? If you mention a virus, we know the size you're expecting, because we need to match our genome and the multiplexing with the platform that we're going to be using, because sensitivity is key. Okay. You know, so looking here, just like for visual, you can see obviously the viruses, they are relatively small, meaning you can actually multiplex a lot of them. And the more samples you can put in a particular flow cell, that means the cost per sample goes drastically down. So it's very important to know what you're working with, and then that will help us decide on what platform to use. The bacteria, they are relatively in between. It's a few megabases there. You know, like you can see Mycoplasma tuberculosis, the TB, you know, it's only 4 MB, but it's a high GC rich organism, meaning we might need to spike more FIREX, and we'll talk about FIREX later on, because Illumina sequencing technology prefers a high diversity library for it to sequence correctly. Remember, we're taking signals. So if it favors certain regions too much, the signal capturing may be affected. but we do offer guidance on that. Okay. Also added here, the yeast genomes, they are relatively bigger. We have the parasite, and a known one is the malaria parasite, Plasmodium falciparum. It's relatively big, so often we're seeing a lot of targeted work, but it's also an AT rich. So that means if you're sequencing only that, you might want to use slightly more FIX, because FIX is a much more balanced genome with almost a equal distribution of the ACGT. Okay. So once you know what you are sequencing and whether you're going to be doing whole genome or the targeted, then that should match with the platform we have. And as you're aware, we have different platforms from small benchtop all the way to high throughput. But here the key thing is, I'm putting those outside because they're coming to end of life in 2029. We're still going to be supporting them, but I want you to just focus on where the Illumina vision is. You can see all the remaining platforms are 2-color SBS technology. Now everything is going to XLEAP SBS because it's performing way much better. And the part I want you to pay more attention to is the maximum read length. So the longest read length we can now perform only on the MySeq 100 is 2x500, meaning if you're doing metagenomics work, you can do V1 to V6. You're not limited to just V1 and V3, which earlier on, we only limited to using either the 500 or 600 cycle kit. Here now, we're expanding the capability of our researchers, okay? You can see the other platform like the NextSeq 2K can also do 2x300 and also on the NovaSeq X as well. So these are new additions with the recent upgrade. The other thing I want you to really enjoy and appreciate is you can see all the older platform, including the NextSeq 550, we needed to do a lot of washes. Now with our flagship platform, they all use patented flow cell. There is no washes needed. That means your maintenance of the instrument is easy. You don't need to do much. You just make sure you remove the data when the run is done. Making your experience with the instrument like less stressful. And another thing with all these platforms, especially these two, MySeq and NextSeq, you do onboard denaturation. So you don't need to do so many steps before you load. Okay. And in addition to that, another big addition is onboard drug analysis, which is available on this platform like MySeq 100, NextSeq 1K, and NovaSeq, whereby you can do your analysis on the instrument, meaning you don't need to have to be like a bioinformatician guru to do this. Just press a button and you're going to get analysis done for you. Okay. So here just showing you the key platform we're focusing on and the different flow cells they have and the maximum reads they can generate. You can see here with the NovaSeq X Plus, we have 25 billion sequencing reactions taking place at the same time. So that's the highest throughput platform we have. You can just think about it. When we talk of like the Saga sequencing forward and reverse, yeah, here we have about 25 billion. Compare that with obviously now our entry level MySeq 100, where you have 5 million sequencing reaction or 5 million clusters passing filter, because the clusters that pass filter are the one we generate the data from. Okay, so the MySeq 100 has the longest read and obviously the smallest clusters passing filter, which is 5 million, but you can it can go up to 25 million with the iSeq i100. The iSeq i100 Plus can add the 50 million and 100 million. Okay, and then we have the NextSeq. It's a little bit, you can see it's a continuous. The highest cluster generated passing filter for MySeq 100 is the entry for the NextSeq 1000 and 2000. Okay, and this one now we just added recently the 5 billion flow cell on the NovaSeq X Plus to just allow our clients to be able to work, be able to manage the projects much better, because the jump from 1.5B to 10B was too big. So we recently added this one and added a 600 cycle kit on the 1.5B. Okay. So what does all this mean? Remember, you need to match the genome size with the capacity of the instrument. And I'm not going to go into detail. I just wanted to highlight where we get the GB from. So for example, if you're running the 500 cycle kit, using the 25 million flow cell. So if you take 25 million and you multiply by 1,000, you get 25 billion. And that's how we end up with the 25 GB. And that's the same concept we apply across all the different platforms. Okay. So based on instrument capability, that's how we are able to offer you a high-level guideline on what application you can run on different instruments. For example, you see the most cost-effective way to do large whole genome sequencing would be on the NovaSeq X Plus. That may not work optimally here. And if I give you an example, if you think of like a human genome is about 3 GB, and if you aim for maybe a coverage of 40x, so 3 times 40 is about 120 GB. So you need an instrument that can generate that amount of data. That's only reasonable viable on the NovaSeq X Plus. And there you can do maybe up to 60 whole human genome, 60 to 64 on the 25B flow cell. Okay. Then if you can't do the whole genome, you want to look at the exome, and the best platform for that would be the in-between one here. Okay. It's not like you cannot run targeted work on these NovaSeq, but it's not cost effective because we are limited by the number of indexes we can use. Currently, we only, from Illumina, you can only do up to 384 unique dual indexes. So meaning that you may not have enough short, a small genome to fill even a 1.5B flow cell. Okay. So just to bring the idea of why it's important to have the right question and to match that with the instrument you're working with. I have given this animation here. I have an individual, an experiment whereby you have somebody who is looking at germline mutation, which I said it's what you inherited at birth from the parental gametes, and it's present in every cell. It can be detected anywhere. And those occurs at high frequencies, And for that, to pick such a germline mutation, you only need 50x coverage. So if you take 3 GB multiplied by 50, you need like maybe 150 GB of data. So that means you can put many more samples that I'm mentioning, like up to 60 to 64 human samples can be sequenced on a NovaSeq X25B flow cell. If you're looking at somatic mutations, these are things that you acquire in life, maybe due to lifestyle or other unfortunate incidences. So they are localized to certain area and they occur at very low frequencies. For that, for you to be able to detect that, you need much more sensitivity. So you need like 10 times more coverage. That means if you have to do whole genome, you can only maybe like do a tenth of maybe like 6 samples per flow cell. So the cost per sample is much higher. So you need to match your questions with, obviously, with the kind of the sensitivity that's required. Okay. So just to give you like a visual understanding, and I must say I had an experience with this not so long ago whereby I wasn't feeling too well. You go to the hospital and they kept coming for blood samples every other day, trying to see what is making me ill, And I tell you, I had one of the most traumatizing experience when they get, the CSF, the cerebral spinal fluid. And I'm like, you have to figure out what is affecting me from whatever sample you have, because there's no way I'm going to accept you for you to take another needle into my back to try and get a sample, because they'll go test with these old technology. And I'm like, don't we have a comprehensive assay, at least like a panel whereby you can look at different things all at once, So it's sad to say, we may say we hear technologically and internally that this is where we're talking about some of the applications we want to do. But when you go to most hospital settings, we're still in the single gene, single assay test, whereby you don't even have enough sample in your body to get the information. At least we should be pushing to get these panels you know, whereby they can test several things more at once. And as we move forward as the cost of sequencing becoming lower and lower, ideally you want to be doing more comprehensive genome profiling, like what you have with the TSO, and you know, where you can look for the whole genome sequencing, or even those instability that affect even our response to drugs as well, you know. And as we're going on, we remember our central dogma is not just about the DNA and the RNA. We need to look at the protein. And just to make you aware, Illumina now can do protein sequencing as well on the NovaSeq X, you know. But the future of everything is you want to do multi-omics. You want to look at more than just one aspect, because diseases and conditions are complex, you know. So hopefully with enough awareness, we can see more of this getting acquired as we get sequencing becoming more and more accessible. Okay. For another short break, and with some few questions here. Of the different Illumina sequencers that I have described, which one has the longest sequencing read length? And if you want to get the bonus point, what's the read length? That's the first question. And the other question is, what is the maximum read length on Illumina instrument NextSeq 1K, 2K, and the NovaSeq X? I think I did emphasize on that long enough. And then the last one is, which Illumina sequencing platform feature integrated onboard drug and secondary analysis, meaning that you can get your data analyzed on the instrument, you know, so the only thing might be left is to look for those like tertiary interpretation. Which platform supports that?
So we have an answer from Malik for the first question. So 2 times 501 MySeq 100 series.
Perfect.
Thank you, Malik.
And the second question, what's the maximum read length on the Illumina instrument NextSeq 1K, 2K, and the NovaSeq X? For the NovaSeq X, that was just introduced recently, but they both have the similar longest read length.
So we have 2 times 301 for NextSeq and NovaSeq X.
Correct, perfect, thank you.
And then for the question on integrated onboard analysis, I think Samuel, MySeq 100 plus.
That's one of them.
Yes.
Yeah, that's one of them. There are two others.
We have a NextSeq 1000, 2000 from Bixa Ogola. Thank you.
Okay, so actually the answers are there. So it's both NovaSeq and the NextSeq and the MySeq 100. But the one that's more comprehensive is the MySeq 100, because even for infectious diseases, we have like onboard analysis for the VSPs, the IMAPs, and small whole genome sequencing. For the NextSeq 2K, 1K, this was more geared for human-related applications. So you'll see the infectious diseases are not there. So you'll have to use either that part, or you can use our base space, because we have a lot of infectious disease analysis free of charge on the cloud. Okay. So, but for things relating to RNA, DNA amplicon, exome, you can do onboard an NextSeq 1K, 2K, and obviously the NovaSeq analysis. Okay. So we do all that to, you know, obviously to serve the different industries as how we introduced. I'm not going to repeat this, but I just wanted to reassure you, like, all those are aimed in serving all these different industries. And I can say, like, one of the more popular one for the reproductive health, you know, the NIPT, you know, haven't seen much happening in Kenya, but in South Africa, it's one of the most popular workflow we support here, whereby at 10 weeks, you know, expectant lady, you can be able to tell, you know, the gender of your child, and if there's any chromosome abnormality, and just from a blood sample, which is quite incredible. So in 10 weeks, you can tell what are you expecting, and you can be better prepared, especially more popular with now that people are starting to bear kids much later in life. So you want to be sure, because we know as the age progresses, the risk of having a neuploidy increases. So that's something that's worth paying attention to. And it's a good business, to be honest, because you know how we are operating recently. And the others are more self-explanatory, but the one I wanted to mention on is the multi-omics. As I keep saying, Illumina is pushing towards this direction where we look at RNA, DNA, and other data from other assays, maybe not sequencing or nucleotide-related or amino acid. You can bring all those together to make a conclusive, full picture of, for a patient, for example, with the multi-omics, including the data analysis. Okay. So, going back to where we're here, as we mentioned earlier, we have different key methods that align with the different workflow. The first one is obviously the whole genome sequencing. It's the most comprehensive. We are able to discover novel variants, even structural variants. And as I said, we have different kits we can use for that, and I'll show you briefly, and the advantages of each, but it can be more costly, if you don't manage to use the right platform. So for example, if you only have few samples and you're running the 25B flow cell on an NovaSeq and you're not pushing at least 60 to 64, then the cost per sample is much higher. If you want to do whole genome sequencing for somatic variants, you can do a fraction of that, like a tenth of that. So the cost is much higher. That way you see now people opt for maybe amplicon or hybrid capture. Amplicon, I normally say, is quickest and the dirtiest. We know it's low cost. Any lab that's doing other molecular biology work can adopt any amplicon work. And I'm talking this for whether it's oncology, inherited diseases, or even in infectious disease, and we have several products under the amplicon. So it's fast, yeah. Often you can achieve much higher depth, meaning the sensitivity is much improved, and very easy to analyze, because if you get data for the entire genome, it's so much data, sometimes even some of the data analysis solution struggle, they take so long. And that way you see Illumina Dragon, is an accelerated way of analyzing. They use more or less similar solution that out there, but they add features to accelerate the process. For some of these analyses, depending on the data you have and the platform you're using, can take days, if not a lot of hours, many hours, but days even. But with the Dragon, most of them, they are very quick because it's accelerated data analysis. The main pain point with the amplicon is you get a high duplicate. Obviously, amplicon, most of them are duplicate. So you have to really optimize the cycles. And here, one key part I can advise you is you'd rather try to improve on your PCR efficiency by increasing, the input, if it's possible, and reducing the number of cycles. Because what we see happening is you may be even using a high fidelity enzyme. Like you saw, Illumina moved from SBS to XClip SBS by improving on the enzyme, the polymerase we use. But the more cycles you have, if by any chance, maybe after a certain number of cycles introduce an error. So if you keep increasing the number of cycles after the substitution error, for example, was introduced, that fake signal becomes part of the signal. So you may not be able to pick, oh, this is actually a processing error. So please try the best you can. All our protocols will guide you and tell you, like, if you have an input of this range, use this number of cycles. The lower the input, the higher the number of cycles. And that also increases the chance of you getting the errors, okay? But that's the amplicon, quickest, easiest, any lab can do. You don't even have to have a very technologically sound technician. Anybody can do that, okay? If you go to hybrid capture, which Cecilia introduced, it's very comprehensive. That's what we use for the exome. That's what we do for viral surveillance and the respiratory panel. And just to give you an idea how robust hybrid capture mechanism is, when we were going through the COVID SARS-CoV-2 pandemic, yeah, we started with Arctic Primer, I think, version 3. Within no time, we're on version 4. As we are speaking, I think we're on version 5. Guess what? The panels that were designed by Illumina for RPIP and RVEC, these are respiratory pathogen panel, infectious, panel, yeah? Those panels were still able to detect other variants of the virus without having to change anything, because they use the degenerate code, so they are more forgiving, but the workflow is not that friendly, compared to the Amplicon, but I would say, it's getting simplified. I'll also introduce some of the changes we have done to the Illumina protocol. The fourth one, obviously, is the metagenomic, whereby you're looking at everything, everywhere. So that one, you're limited by the sequencing instrument capacity. So metagenomics are best done using our highest density platform like the NovaSeq X, 25B, 10B, or on the NextSeq 1K, 2K, mainly like P3, P4. Those one are best for the metagenomic work. Metatranscriptomic, maybe not as much data is needed, just looking at the RNA, but still you need a significant real estate. Okay. So you'll see there are so many kits, and I'm not gonna go into details about this, but I just wanted to give you like a feel of what we have and the options we have around. So for DNA, I'm just highlighting like 3 here. If you go to the publication, the most popular workflow is the Nextera XT, but it's not the best performing, because sometimes as researchers we believe what is in the print. They'll say, hey, I want to buy this, but this was a good introduction, but it's so limited in that you can only work with 1 nanogram of input. If you put more than 1 nanogram, you end up fragmenting your DNA. If you put too little, you end up overdoing it. 1 nanogram is a very small number because you're working with very little, so it's very difficult to optimize. I always say Nextera XT gave me the gray hair you see today, because it was a very challenging time to be an application specialist, because clients left, right, center were struggling. So we did a major improvement on it by taking the same transposase enzyme, but attaching it to a bead. And that's how Illumina DNA Prep was introduced. This one, it can also accommodate low input as low as 1 nanogram, but goes much higher up to 1,000. But it's much more robust. We get even distribution of fragment sizes. Remember the insert on our library? Here it's much more controlled. That way we say, for example, Illumina DNA Prep, we don't need compulsory traces, because we know we end up always with about 600 base pair library. Okay? As you know, anytime you have, this has a PCR step, as Cecilia explained. Anytime you have a PCR free, there is a bit improvement. That way you see Illumina DNA PCR free is the one we opt for, that genome, because we can see a change in the coverage you get in the biasness. But for small genome, Illumina DNA Prep, including the PCR aspect, doesn't affect things that much. Okay. And then for the targeted, we have several solutions, whether it's Illumina DNA Prep with enrichment, same kit, but we just use an enzyme that cuts much shorter fragments so that we are able to pull them using the hybrid capture. Okay, so this is what we use for exome and also for urinary pathogens. Okay, so a set of them for that. And then also we have the Pillar OncoReveal. This is used for cancer and also for hereditary diseases. And we recently, not so recent, we modified the COVID-seq to get the IMAP, which is brilliant because we get all the components. You can put RNA, you get it's converted to double-stranded DNA, and you can proceed with the work. So this one is quite popular, for new pathogens, and it's also amplicon-based as well. And for the RNA, we have different solutions here, whether it's Illumina stranded total RNA, which I said you get looking at there, or the RNA coding and uncoding. Quite interesting for new organisms that are not well annotated, because you're able to get much more information and see the gene expression levels. And then we have the messenger RNA, stranded one. And Illumina now has just introduced a new small RNA, microRNA kit. So this has replaced the old TrueSeq, And then for targeted for RNA, we have Illumina RNA prep for enrichment. This is the backbone for all our viral, like the surveillance panel, the RP, VSP, and also the VSP even version 2. All of them are based on this. But we have done major upgrade whereby all these RNA kits, now we have added the rapid workflow, which are much shorter, and you're able to do even the enrichment workflow within a day. Okay. So with all that, so this is kind of like a bigger picture of all the solution we have for infectious disease. The COVID-seq are still there. Obviously, we have the Dplex. This is a collaboration with GenoScreen for TB, which is WHO endorsed. We have the IMAP, and we have several versions of the IMAP. IMAP flu, IMAP mpox, IMAP chikungunya, IMAP. There's a list of them. You can also access that from the website. And for the enrichment panel, as I mentioned, the hybrid captures, we have the RVEC, the VSP, both versions, but I don't see the reason why you should go for the version 1, because the version 2 includes 2 added viruses compared to the VSP one, which only had 66. And all these, you are able to do like on the MySeq 100, you can do most of the analysis excluding the Dplex, because all these are already on board the instrument, including the drug and microbial enrichment plus, which you see is what we use for all the hybrid capture protocols. Okay. So quickly, just to conclude here, a high-level overview. So if you're looking for the whole genome or types of DNA, we need to be careful and choose depending on whether it's large genome or small genome. You can see for the large genome, the minimum for Illumina DNA prep is 100 nanogram, but you can go up to 500 nanogram. Okay. For what we recommend for whole genome for large genome is the Illumina DNA PCR free, and you can see that one requires a minimum of high quality DNA, at least 25 nanogram. We have added 2 more solutions, the 5-base, whereby you look at the sequence and also the methylation pattern in the same assay. Okay, so this is a new kit, and obviously we have been mentioning a lot about Illumina TruePath genome. Okay, so for small genome, stick with the Illumina DNA prep. We have a lot of projects going on this and getting really good results. It's a very quick protocol, not as fast like the PCR free one because of that PCR step, but it's quite robust and you get consistently more or less the same library length, about 600 base pair. My advice is, whenever you see a kit saying 25 to 300, don't aim for the low minimum. Aim for anywhere in between to the maximum. Often people take the average and they're like, you know what? I wonder if it's possible for small genome, the bacteria pure culture, you can easily get up to this amount if using the correct kit. So aim toward the highest, higher end. For example, we say you start with 30 microliters of your solution, So if you take 500 divided by 30, you see you need any sample that's showing you about maybe 60 nanogram and above, maybe 20 nanogram per microliter, then you're good, because then you're well within that range. Okay. If you're looking for the targeted, we have several solutions, whether it's hybrid capture, we have the TrueSight options, and for the hybrid capture here, the more popular in the TSO 500, because you analyze 500 different genes and also RNA as well. And then for exome, we're using the Twist version 2.5. And for the amplicon, these are the options. The more popular one is the OncoReveal from Pillar. And once again, if you're looking for not just the DNA, you want to see what is active, then we have the RNA option, which we discussed earlier. And the new addition to this is obviously the single cell transcriptomic, Illumina kit, yeah. And then we have the StrataMap, spatial transcriptomic. You have a tissue and you're able to analyze the RNA expression. Okay. So I would like to stop there because the other slide is just talking about the changes we have done with all our RNA kits. So we have given it a new facelift, to make the protocols much quicker. You can do it even within a day. And we have also adjusted the input requirement from 1 nanogram. Initially, it was only 10 nanogram was the lowest, but now we can go as low as 1 nanogram. And these new protocols are much friendly. They are, whether it's for total RNA, messenger RNA, or targeted enrichment, which we are talking about for the infectious diseases. And just a reminder, you have a maximum of 384 unique duo indexes, because I see there's a lot of scientists here who are keen to understand how did Illumina manage to come up with this rapid RNA kit. So I have just put like a comparison. This is the new kit where we have combined a lot of steps into one step, the cDNA synthesis and the fragmentation. So all these 3 steps we used to do separately are all grouped here. And remember, when you're on the bench, the fewer pipetting steps you have, the better. There's less room for errors there. So looking at that, you are able to finish, including enrichment, all the work within a day. I have done a lot of RNA with enrichment, for the VSP and the RPIP, and always used to do it at least in 2 to 3 days because you have an overnight incubation and all these steps. So you don't want to scare somebody, even when you are starting, you don't want to start with the enrichment. As I said, it's a little bit more involving. It's not difficult, but with these changes that have been done, anybody can do that. Okay. And that's just like the graphic representation of the whole changes and what you can do. So we removed a lot of cleanup steps to make it more user-friendly. Okay. So as I take a small pause here, as you have seen, there's so many solutions we have out there. And sometimes you may not know where to start. So we have very nice solutions Illumina has put together, that helps you to decide on which platform or which kit to use, So I don't know, Cecilia, whether we should take a break here or should I demonstrate how to use these 2 tools? I don't want to extend this for longer than I have. Any questions?
We wind up.
Okay. Okay, so I'll not go into detail because they're more self-explanatory. Maybe if we have a moment, we can go through that. But otherwise, because we're going to share these, I'm happy to. The link is from the Illumina website, and you just get asked a few questions, and you're able to narrow down to a certain kit.
Yeah, we will share the resources, and then the participants can play around with it.
Okay. Okay, cool. Yeah. Let me stop there.
All right. Thank you. All right, so let me share my slides. Thank you very much. You can see my slides? Yeah. All right, sawa. Let me go to present. Okay. Sawa. So just as we wrap up, because time is much spent, just to recap, thank you, Charles, for that comprehensive journey through. I think at every stage you touched on the different things that one would consider when thinking about how to answer their question, their research question on the entire workflow, not just along the library prep. And maybe just to emphasize something that you touched on is that for each workflow that has been mentioned, there's usually a guide that is provided by Illumina that the researcher can follow, and it highlights the sample input recommendations. So when you have extracted your DNA and you have done your QC and you know your DNA concentration, you already know where to begin. So the different concentrations of your samples and if it meets the criteria for input into the workflow that you're about to undertake. There's also a lot of tips within the reference guides on how to handle your workflow, how to avoid cross-contamination, how to seal the plates when you're taking them into the thermocycler to also ensure that you're not experiencing any evaporations, you're not having, again, cross-contaminations. And of course, your thermocycler needs to be compatible to the consumables that you're using. There's also tips about how to handle your different reagents, especially reagents that have specific handling techniques, like the BLTs that are used for the tagmentation steps, the wash buffers that are used within the workflows. So it is important to read through your guide before you go to the bench. You don't just go to the bench and start at take your DNA and start making your master mixes. It is important to look this through the tips to understand how to handle your different stages. Then, of course, for those who are using the MySec, as Charles has mentioned, there's a denaturation outside of the instrument before loading for the sequencing. You definitely get a guide on how to use your to do your denaturation and the best practices. So for each system, there are guides on how much concentration of your library to load. So as we emphasized on the QC, you do your quantification, your fragment analysis, and then dilute your library properly to the right loading concentration so that you're able to get the right data. And Illumina provides the different tips for the different stages of your workflow for how to achieve it correctly so that you're not guessing and you're not adding or reducing the different concentrations or stages that you're supposed to be doing. So I'll change gears a little bit here and just would like to hear from you, from people who have worked within these workflows. And to kick us off, I think I've had a few conversations. I've been on the bench with some of you, and we've experienced quite a bit of interesting modifications, or let's say we've had to sit through brainstorming around how to structure maybe different workflows to get the best results. And maybe I'd like to invite some of our participants, and I'm gonna ask to be allowed to single out a few of you so that we can maybe get a bit of your perspective on your experience around workflow. So I'll start with Elikaya. I hope he's in the room. If you could mute yourself and maybe you could give the audience maybe some advice around maybe what was your biggest challenge in establishing the workflow that you're running, and if you were to give one piece of advice to people who are starting the NGS, what would it be? Can you can unmute yourself. You have the permission to unmute yourself and speak. So this one goes out to one volunteered individual, Elikaya. Let's see if he's in the room. Another person who I do know that we've had quite a bit of workflow specifics with, Samuel Shenge. And for you, I think one of the biggest lessons that we have with you was around library QC. So maybe you could share a bit about which control steps had the biggest impact in your results around the workflows that you run. Samuel, you also are able to unmute yourself.
Okay, so thanks for giving me this chance.
All right.
The biggest impact is when you are seeing the indexing step. There was some this technology where you use QSEP and you could be able to determine the fragment size. And I noticed if you have an amplicon and you do indexing, you expect your fragment size to increase, or the average fragment region to also kind of increase. And that's what kind of determines. And the in terms of pipetting, the accuracy, I know I've been using MySec, partially MixSec, but I know the the pipetting skills are the major determinant of your results. Yeah, thank you.
All right. So maybe you'd have told us what you were doing, if that's okay, so that we can relate to the stages of QC.
Okay, so I don't know what to take because I've done both HLA typing, microbiome, and transcriptomics. So which one do you want me to go with?
Up to you. So the key is maybe to understand any advice for any people using the NGS workflows, and what are the areas that for you have been important in library prep to pay attention to because they have an impact on your results.
Okay, so I'll let me just go general. So first, you have to make sure you have gone through all your SOPs and to know and you may ensure that you are setting up all the reactions at the required temperature. You don't want to find yourself like you are supposed to do a cleanup step. Maybe you have a short time period between the fragmentation and adapter ligation, and you find that you forgot to thaw your cleanup bits. So I did know, and then there are some steps where you are given the safe stopping points. So you have to ensure that you do not step in a safe stopping point. For example, an assay like HLA typing, you'll find that immediately after fragmentation, you must add the ligation mix without cleanup, which means you should have prepared your ligation mix earlier and kept it on ice. So you delay on that, and then you find problems later on. And then in terms of the library normalization, it is best for when you are doing sample normalization, it's best you do the normalization on the day that you are doing the pooling, and if given a chance, you sequence it on the same day. Thank you.
Thank you. Thank you for sharing. As we wait for Elikaya, maybe Charles, we can answer some of the questions that we have on the chat. So we have a question around the true path. And the question is, can you use RNA or protein as your starting material for true path?
Okay, yeah. So currently, the only option is DNA. You cannot use RNA or protein. And it's actually, as we're speaking, it's only for human DNA, but it's new product. So maybe with further development that might change. And I think maybe a better way to look at this is we can go to the website and have a look at the description for it. I think that was, okay, you have some slides. Yeah, so because also I saw another question like, how does it compare with Illumina DNA PCR free? You know, I would say like it's like totally different worlds here because as you saw, this is the only protocol we have that does not start with library preparation. And I remember when Illumina were introducing it, we all kept saying like, how can you sequence without having any library prep, you know? So what we aim for is obviously you try and introduce high molecular weight DNA into the flow cell. And because those flow cells, they are patented flow cells. So we have nanowells all around and we know the distances. And just as you'd expect, you know, if you have a very long intact molecule, it's more likely to be within certain nanowells within the flow cell. So chances are even when we segment it within that area, we know this strand was initially one, and then using the proximity knowledge, then you're able to put back the genome together. That way we're saying it's not like typical Illumina sequencing in that we are able to get a more comprehensive genome, even areas that maybe the enzyme may not be able to cut it as nicely. With this one, you're able to overcome that challenge. And the other true beauty of this is the reduced pipetting step. You can see it's only 5 steps. You add water, reagent 1, 2, 3, and you are ready to go. Okay. Muted. We can't hear you.
Oh, okay. So let me stop sharing. So essentially this workflow, as Charles has mentioned, is running on the NovaSeq, and it is able to sequence the human genome without the library prep step, and yeah, give you long read insights from short read sequences. We don't know if Illumina will get around to having no library prep in other systems, but for now, it is running on the NovaSeq system. Then we have a question around what is the impact on sequencing if you have low concentrations like 1 to 5 nanograms versus 200, 500 nanograms for Illumina DNA prep kits. Charles, again, you'll kindly take this one.
Okay, yeah. So, as we have put it officially, you are able to work with both ranges from 1 nanogram all the way to 1,000. But what I have seen is, yes, we do have even some graphical, if I was opening the slide for Illumina DNA prep, whereby you aim for anything from 100 nanogram and above because you end up with kind of like normalized libraries, meaning you don't need to actually go and quantify every library at the end of it to know how much you have, because the bead only has a limited surface area. So when you reach its saturation, there is no more DNA that can attach, and that allows us to achieve normalized library across the board. Okay, so when you go for these lower range, as I'm saying from experience, when you go to the lower range of anything, even a pipette, you'll see like you have a P10 and starts from 1 to 10. If you try to use that one to measure like one microliter, it's not the most precise. So the same concept applies. The lower range tends to have a lot of variation. So you want to aim, if it's possible, obviously the sample materials you have, that's what dictates what you can work with. We get a lot of inconsistency with that lower range. That's why for me, I prefer mid to the upper range if possible.
All right, and then we have 2 more questions. So one is to clarify what determines the percentage of phi-X that you spike into your library. And the other one is, if you have variable concentrations across your samples, do you recommend standardizing the input for library preparation to the same concentration? Or do you recommend using different input concentrations and then sequencing them separately?
Okay, so regarding the phi-X, yeah, remember the phi-X, normally if you look at most of our protocol, Illumina will say it's optional. But for me, I say it's compulsory for every run to have some phi-X in it. The question is like, how much am I putting? If you're doing whole genome sequencing, for example, of a bacteria, you expect a lot of many variable random fragments of different sizes, different composition. There you can go to the minimum pipettable amount. So 1% can do, right? If you're sequencing, let's say like amplicon, especially 16S, which is kind of like a lot more popular, especially the MySeq 100 because of the increased read length, we're seeing a lot of that. Those ones, they are more similar than different, meaning because there is not enough diversity, and we can show you from an SAV sequence analysis viewer data, you can see like it looks extremely spiky, For that, based on the platform you're running and the control software you are on, that will decide whether you'll go for maybe like 5% or 10%, because what we do is often, and this is something that I would encourage anybody with the Illumina sequencing platform to make sure that it's running the most updated software, because sometimes we are able to reduce we don't want you to sequence 5X. 5X is going to take the real estate. We want you to sequence your samples more, So you want to try and make sure that you're running the most updated software possible. And depending on what you are sequencing, that is what will determine how much phi-X to put, whether it's the low range 1% or maybe 5 to 10%, depending on the software you're running. Okay. The second question regarding if you have samples that are variable concentrations, that are, I chose a number, So for example, we'll say with the Illumina DNA prep, you'll see, we say and I'm a microbiologist, so I'm always biased. I speak more of microbes, yeah. We say 1 nanogram to 500, you know. But if I'm starting this experiment, the easiest way is I'll go for the highest amount. I'll say I need 500 nanogram, okay? So what volume am I working with? 30 microliter. So if you take 500 divided by 30, what do you get? This is a time when I'm even doing a training until you can use your phone. I can't do maths when I'm seated. So you need about, maybe it's about 16 something, yeah? Let's say for the, yeah, let's say it's 15, you know? So any sample that's 15 nanogram per microliter, if you take 15 multiplied by 30, that's giving you 450, you know, nanogram, which is quite within the range. So what I'll do is, if all my samples are like maybe more than 15, I can normalize everything to that, and then I know I'm working with the standard volume all the way. Sometimes you find some cannot even make it to the 100 nanogram. Okay, so those are the samples you want to work on, where you know if it's less than 100 nanogram, you'll have to quantify every sample individually before you pull them at the end of the protocol. So I like to group them. I say this one, it's Monday, I don't want to work, I want to get the run going. So I'll pick anything that's above my threshold. Anything that's above 15, that's what I'm gonna put together. Then these other ones, when I have the momentum for the week, then I can prepare for the second batch, which I know I'll need to spend much more time quantifying them individually. I think, was there that question? If there was, I forgot.
No, there was 2 questions. Thank you.
Okay.
If there are any other questions in the room, you can put your hand up and we can address, especially if you're struggling to make a decision. I think we'd be able to help you there. So as we wrap up, I don't want to go too much above the time. We had said we'll end at 12:45. So just to wrap it up, I think for the key for this session was for us to maybe just highlight that library preparation is a very important determinant of your sequencing success. So it is very important to pay attention to the decisions you make pre and during and post library preparation. First, decisions around, is this the best library workflow for the question that I'm trying to answer? 2, what kind of sample do I have? And then how does that influence my choice of extraction, my choice of library prep as well? And then what are the key things that I need to do to make sure that one, I'm running the workflow as it is supposed to be? What are the most critical checkpoints in terms of QC to make sure that we are loading the right concentration and volume of library for our run? 2, quality control, of course, is very important. As Charles has mentioned, you will end up QCing all your libraries towards the end before pooling. So that will inform you, even when you look back at where you started at your sample. If you had a sample that you started at 10 nanograms, another one at 50, somehow you can correlate the kind of outcomes that you get. So internally, even as a lab, you're able to kind of build those thresholds that are cut off for your library prep, especially on specific workflows. So of course, you have to consider the end-to-end, not just library prep. It is part of the workflow, an important part, but you have to consider the end-to-end as you are thinking about the data that you will get at the end. And I don't know if there are any hands up for questions or any other questions in the chat, but you can reach out to F&S Scientific. Peter and I are available to answer any questions. You can send an email, you can call us, we're able to answer the questions. And we work closely with Peter and the Illumina team to also be able to support you. So unless there are any other questions, thank you for joining us. And Charles, maybe you can wrap this up for us while I leave those contacts up there for anyone who would like to grab them.
Thank you, everyone. And, I always say we like Liverpool, you'll never walk alone, So we may we may be at different places, and we're here to represent. So if you need any assistance, you can reach out. You see our activities on the social medias. When you meet on the street, say hello. But all the best in everything that you're doing as well. And thank you for your time. Thank you.
Right, thank you.
Kwaheri.
Okay, bye. Thank you, Charles.
Thank you.
Oh, there's a question on the chat. Yeah. So Prudent, I don't know if you're still in here. Yes, you are. Let me drop my number here so that you're able to reach out to me with this question. The capacity of the flow cell, if you want to multiplex how much space you have, I can still answer it. So we all the different flow cells can run a lot of samples, up to 384, really depending on the depth of data that you need. So what informs how many samples you put together is how much data do you want per sample? And then there are different flow cell sizes depending on the instrument that you're working with. So we have the smallest instrument, the ISEC, and we have the biggest instrument, the NovaSeq. So for example, on the NovaSeq flow cell, the biggest flow cell of 25 billion, we can run up to 64 full whole genome sequences at 30x coverage. So really what informs your pooling and multiplexing capacity is how much data do you want from the samples. Yeah, so I'm glad you stayed for me to answer that question. All right. So thank you so much and have a great afternoon ahead. Thank you.